Imagine um mundo onde você possa contar com gente que se dedica a fazer o seu mundo melhor. Gente que está presente quando você precisa de apoio, de confiança, de motivos para acreditar. Imagine contar com pessoas que te estendem a mão diante de seus maiores desafios e que se alegram com cada uma de suas conquistas. Pessoas que te compreendem, porque compartilham com você os mesmos ideais e a mesma missão. Pessoas que pensam além, por você, e que estão sempre atentas aos novos ventos, às mudanças inevitáveis, às estações que se sucedem, os engenhos do tempo. Pessoas que reduzem suas dúvidas e multiplicam suas conquistas. Pessoas que sabem que a vida não é só o vai ou o vem, mas o vai e vem que nos leva para o lá dos nossos planos e para o cá da nossa realidade. Imagine contar com pessoas que te ajudam a transformar o não em sim, as incertezas em possibilidades, os desafios em novos caminhos. Pessoas que caminham ao seu lado, mesmo quando o mundo impõe novas e inesperadas barreiras. Essa gente parceira, flexível e humana, atende por um nome: FTD Educação.
Conte com a gente.
Conte com a gente.
Conte com a gente.
Conte com a gente.
Conte com a gente sempre.
Good morning, everyone. We're starting now the activities of the oral presentation session of the Institutional Program of Scientific Scholarships, PIBITI, PIBIC, and PIBIC Jr. at PUCPR. This section will have study presentations by students from health sciences. Our dynamics will be as follows. Each student has 10 minutes to make their presentation, and then they will be questioned by 2 examiners for 4 minutes, and so on. There will be 8 papers presented in this section. I wish everyone good luck, and let's start by calling the student Marjorie Luisa Barbosa, supervised by Professor Alexandra Cristina Senegalia from Med School, PIBIC with scholarship from Araucaria Foundation.
O que é isso?
Rafaela.
Marjorie.
Eu cliquei errado.
Oi, estão me ouvindo?
Yes.
Hello, everyone. My name is Marjorie. My undergraduate research project has as supervisors Alexandra Cristina Senegaglia and Letícia Fracaro. The title of the research is Validation of a Protocol for Isolation and Expansion of Umbilical Cord Tissue-Derived Mesenchymal Stem Cells. Before I present my project, I will talk a little about myself. My name is Marjorie Luisa Barbosa. I'm 23 years old. I am a computer technician by TecPUC, and I am a biotechnology student since 2017. This research that I will present today is my second project in the cell therapy field. My goal is to study abroad and become a scientist in this field. PUCPR Core for Stem Cell Technology is located at PUCPR campus. It develops basic, preclinical, and clinical research. It is important to highlight that the core is licensed by Anvisa that allow us to infuse mesenchymal stem cells into human beings. The research lines of the core include autoimmune disease, cardiology, hematology, neurology, orthopedics, and pneumology. Well, mesenchymal stem cells are undifferentiated cells that are capable of maintaining their undifferentiated state or differentiating to other cell lineages. Mesenchymal stem cells also present paracrine and immunomodulatory activity and low immunogenicity. That's why they present an important therapeutic potential. These cells can be obtained from several sources, including umbilical cord tissue. Umbilical cord tissue-derived mesenchymal stem cells, also known as UCMSC, have the potential to inhibit apoptosis and inflammation, increase revascularization, release several trophic factors. One of the advantages of this source is a non-invasive collection procedure because the umbilical cord is usually discarded after birth. Mesenchymal stem cells used in these studies need to be complied with the ANVISA regulations to be validated and subsequently infused into patients. That is, all the procedures need to be validated. However, to assure the use, the quality, and the efficiency of the cells into clinical studies or posteriorly as an advanced therapy product is necessary to perform several assays. Thus, a standard protocol is required to assure the safety of the future patients. Therefore, the aim of this study is to validate a protocol for isolation and expansion of UCMSCs through several experiments. This study is approved by the PUCPR Research Ethics Committee. Initially, 5 samples of umbilical cord were collected after the signature of the informed consent form. Then the samples were washed and dissociated mechanically and digested enzymatically, and then plated in a culture flask with IMDM medium and fetal bovine serum. After that, the cells were submitted to several experiments, including immunogenicity, karyotypic analysis, immunophenotyping, colony-forming unit fibroblasts, and cell differentiation. To immunogenicity and immunophenotyping assays, the cells were marked to specific antibodies, each assay with different antibodies, and then the samples were analyzed by flow cytometry. 7-AAD was used to analyze the cell viability and annexin 5 to indicate the cell in apoptosis. According to the International Society for Cell and Gene Therapy, mesenchymal stem cells must express the antibodies in green and lack expression of the antibodies in red, as you can see in these tables. Cell differentiation. Cells were separated to each differentiation experiment, cultivated under a specific differentiation medium during 21 days, stained and quantified. Adipogenic differentiation cells were cultivated, stained with oil red O, which stained the lipidic glucose. Then the differentiation was quantified by spectrophotometry. Osteogenic differentiation cells were cultivated, stained with alizarin red, which stained the calcium deposits, and then the differentiation was also quantified by spectrophotometry. And chondrogenic differentiation cells were cultivated and then stained with toluidine blue. Colony-forming unit fibroblast assay was used to characterize the UCMSCs. Sample under different concentrations, 800, 1,000, and 1,500 cells were cultivated with IMDM and fetal bovine serum during 14 days. On day 14, the cells were stained with violet crystal, and the colonies were counted through a microscope. It is important to highlight that 50 or more grouped cells is considered a colony. Karyotypic analysis by G-bending was performed to evaluate chromosome abnormalities. In short, the cells were stopped methodically, stained by GIMSA, and the karyotypes were analyzed by microscope and software according to the International System for Human Cytogenomic Nomenclature. results. Regarding immunophenotyping that is represented in orange, the antibodies are in agreement with the standards established by the International Society for Cell and Gene Therapy for mesenchymal stem cells characterization. Immunogenicity, represented in purple, show a reduced expression of CD40, CD80, CD86, and HLA-DR, and a positive expression of HLA-ABC, showing that UCMSC can be used for allogenic transplantation. All the samples of UCMSC were differentiated into adipocytes, osteocytes, and chondrocytes. Here we can see the lipidic vacuoles that indicates the adipogenic differentiation. Osteogenic differentiation can be seen through calcium deposits stained in red, and chondrogenic differentiation can be seen by the proteoglycans in the cartilage matrix. This demonstrates that the cells are mesenchymal stem cells and are in accordance with the International Society for Cell and Gene Therapy guidelines. Regarding quantification of adipogenic and osteogenic differentiation, we can see here, comparing to the controls, that the samples were differentiated. The colony forming unit fibroblast, the colony forming unit fibroblast assay presented an average of 20.8 colonies, and the average colony formation efficiency was 2.08%. This demonstrates the presence of mesenchymal progenitor cells. Here we can see the colonies in this first picture stained in purple, and one colony microscopically at this picture. Furthermore, the karyotypes did not show any chromosomal abnormality, demonstrating that the samples did not show genetic instability during cell culture and can be used in cell therapy. Final considerations. All the samples can be characterized as mesenchymal stem cells since they present results in accordance to the International Society for Cell and Gene Therapy, as well as accordance to several studies. Cell therapy regenerates tissues and organs. Thus, studies like this assurance the safety, quality, and efficacy to the use of the cells in several studies and treatments. Finally, this study was essential to validate the quality control procedures of the advanced therapy product. I would like to thank my family, especially those who are in this picture, my friends too, and to the Core for Stem Cell Technology, especially to Alexandra, Carmen, and Letícia, to Fundação Araucária and PUCPR. Thank you for the attention.
Okay, thank you, Marjorie, for your presentation. It was just on time. Professor Andrea, do you want to start, and then I can do it if we have time?
Okay, thank you. Okay, Marjorie, thank you for your presentation. It was amazing. I have maybe one question for you. The mesenchymal stem cells is widely used also both clinical and also in basic research. And you show us that these cells can be used in medical or clinical practice. My question is, what is the relevance for you the validating a protocol for isolation and expansion of these cells in this context that these cells is already used in clinical practice?
To assure the safety, the quality, we have to present results according to the resolutions from Anvisa. And we have to present also this characteristic, safety, efficiency, quality, because if we don't, the patients could have some issues during the infusion, maybe tumors or contaminations. So I guess it's extremely important to do research like this.
So in the research or in literature, we don't have these protocols or these validation protocols right now. You made this to organize all the assays that it's relevant to use these cells in clinical practice.
Yes, but not all of the experiments that we have to present to Anvisa. It's some
Okay.
experience.
Okay, I understand. Thank you again, and congratulations.
You're welcome.
Okay, congratulations, Marjorie and Professor Alexandra, which supervised the study. It's a very relevant study scientifically and clinically, as Professor Andrea had already said. I have only one question, which is more a curiosity. You used only 5 samples to validate a protocol, which is established protocol already. And do you think a wider sampling would influence your results, or this is just the standard and you don't have to do it with more samples than 5?
I think to undergraduate research, 5 samples, it's okay. But I'm sure that to Anvisa, we have to present more than 5 samples.
Okay, and maybe if you
To
intending
assure
to publicate as well, for a publication as well, maybe?
Yeah, I hope so.
Okay, congrats again for your brilliant study
Thank
and
you.
presentation.
Thank you.
Okay, so we can move on for the second presentation. The student is Fernando Arfux Maluf, and the supervisor is Professor Carlos Eduardo Coral de Oliveira. Are you ready, Fernando?
Yes, I am.
Okay, you can start.
Okay, good morning. My name is Fernando, and today I'm going to present the results of my research entitled Influence of Probiotic Supplementation on Gastrointestinal Complications in Colorectal Cancer Patients. It's a systematic review with meta-analysis, and it was supervised by Professor Carlos Eduardo. Colorectal cancer is a highly prevalent disease. It's considered the second in mortality and third in incidence among all the cancer types around the world. Its treatment involves high morbidity. GlobalCan estimates that around 1,800,000 cases and 900,000 deaths during the year of 2018 attributed to it. Its incidence only tends to grow, with an estimation of 2 to 5 million cases annually until the year of 2035. Currently, its treatment involves surgery and/or chemotherapy, and just like the disease itself, it's related to many possible complications, such as the need for colostomy, diarrhea, bloating, fever, infections, and other complications that can significantly impair the quality of life of the patient ongoing cancer treatment. Although there are many non-modifiable risk factors, such as race, gender, and family history, there are many modifiable risk factors. And many of these risk factors are closely related to the microbiome, such as diet, alcohol consumption, and even smoking. This close correlation led to the hypothesis that by manipulating the microbiota using the probiotics, we could lead to local and systemic changes that could improve the quality of life of the patient ongoing cancer treatment. Therefore, this review aimed to evaluate the influence of probiotics on gastrointestinal symptoms and the duration of the pyrexia on colorectal cancer patients. Our study protocol was registered in the Prospero platform, which is an international platform for systematic reviews. And to ensure the methodological rigor, we followed the PRISMA guidelines for systematic reviews. Our research strategy involved searching for randomized controlled trials on the databases PubMed Medline, Cielo, Biremi, clinicaltrials.gov, and the Cochrane Library, published from the year 2000 up to 2021. We used the keywords probiotics and colorectal neoplasms, and using this strategy, we found 118 articles, which were further analyzed using our inclusion and exclusion criteria.
Bom dia, querida. Muito obrigada. Desejo
Inclusion criteria involved studies that were conducted in humans, published in English, Spanish, or Portuguese, and that involved at least 2 weeks of treatment. And we also wanted studies with a similar PICO to the one of our interest. PICO stands for population, intervention, control, and outcome. And ours was a population of patients diagnosed with colorectal cancer. The intervention that we wanted to evaluate was the inclusion of probiotics on the current treatment of the patient. The control group was composed of patients that had the diagnosis of colorectal cancer and that did not use probiotics. And the outcome that we were looking to evaluate was whether this intervention could lead to a reduction in adverse events associated to the disease and to the disease treatment. Exclusion criteria were studies with under 20 participants in each group, studies with a different PICO from ours, studies with a high risk of bias, and studies that included other interventions in addition to the probiotic supplementation that could compromise the evaluation. And so as we can see in the flowchart, from the 118 articles we originally had, after inclusion and exclusion criteria were applied, we came down to 8 articles. These articles were analyzed according to their risk of bias using the Hevman software. We found an overall low risk of bias among all of the articles included. We also found a low heterogeneity, which is why we used a fixed defects model for our statistical analysis, and a low publication bias without any asymmetry in the funnel plots during the Eggers tests. The main characteristics of the studies were compiled into a table, which is our table 1. The total population of the patients was of 765 patients. All of the studies included placebo for the control group and some level of blinding. And it's important to highlight that 6 out of the 8 articles included were conducted in the southeast of Asia. The most common strains used in the randomized controlled trials included were Lactobacillus acidophilus, Lactobacillus plantarum, and Bifidobacterium longum. For the meta-analysis, we included outcomes that were evaluated by at least 3 articles, and they were incidence of diarrhea, incidence of bloating, and the pyrexia duration. For the incidence of diarrhea, we found an overall reduction in the intervention group with an odds ratio of 0.40 with statistical significance. For the incidence of bloating, we also found a reduction in the intervention group with an odds ratio of 0.47 with statistical significance once again. And for the pyrexia duration, we found a standard mean difference of minus 0.48, which points for a reduction in the pyrexia duration in the intervention group. And we also found statistical significance for this outcome. When we compared the outcomes that we evaluated in our statistical analysis with the ones of other meta-analyses that included the same outcomes, we didn't find any significant discordance in the literature. However, our meta-analysis was the first one to find a statistical significance for the reduction in the pyrexia duration in the patients of the intervention group. As we can see, both of the meta-analyses that evaluated this outcome pointed to a reduction. However, both of them had a p-value superior to 0.05 and therefore didn't have statistical significance. In the end, we conclude that probiotic supplementation reduces gastrointestinal complications and pyrexia duration in colorectal cancer patients, since the patients that received the probiotics had a lower incidence of diarrhea, abdominal bloating, and lower duration of pyrexia. The main limitation of our study was the lack of randomized clinical trials that evaluated the population and the outcomes that we were interested in. And we feel like we would benefit from further studies that aim to evaluate what is the best strain combination of probiotics and their posology to optimize the benefits that we found, as well as to evaluate what is the best duration for the supplementation. Finally, it would be interesting to understand whether these benefits are exclusive to colorectal cancer patients or if they can be extended to the general population. Thank you very much for the attention.
Okay, thank you, Fernando. Your time was 7 minutes. so we have more time for questions. I'm sorry. Too much to deal with. Okay. Can I start this time, Andrea?
Yes, of course.
Okay. So first, congratulations for this relevant study, Fernando, and your supervisor, Professor Carlos Eduardo. I think systematic reviews are increasing their potential since last year. It was, of course, a source of incredible data, but since COVID and the pandemic, this is being a lot of researchers using this method for bringing some important research data for a lot of med and dental research as well. So I have just maybe 2 or 3 questions only for you, and maybe some maybe some of them you already talked about, but I read the abstract and it calls my attention. So you said that you had 118 articles in your search, and 8 were used for the meta-analysis, okay? Okay. Do you think the limitation of time that you used had some kind of restriction for the studies included?
Actually, the main exclusion criteria was an incompatibility among the population and the outcomes evaluated by the other articles. And so I don't remember the exact number of studies that were excluded because of the time that we established, but it was definitely not the main reason for exclusion of the articles. I believe that we found in the literature on similar meta-analysis that this intervention time was important because the adverse events and the full potential of the treatment wasn't much prevalent before this timestamp that we established. And so I feel like we could trust the results that we could bring more if we established this time.
Okay. Maybe when you're going to publish your paper, some reviewer will ask you about this limitation from 2000 to 2021, but you will answer quite well. Okay, and then just a curiosity again, but studies from Lee, which are in the meta-analysis plots and tables, they are the same study with different follow-ups?
They are studies that involve a different population, and that's why that's the main reason why we included all of them. And so there's obviously a limitation there. We can have similar population considering the geographical region, but the studies were conducted in different patients. We made sure to confirm that, and so the results can be interpreted separately. Okay,
good. And the last one, you said, again, just a correction or maybe another way to write it. You said that the risk of bias was evaluated using criteria from the review manager, the RevMan. But maybe you should say that the risk of bias was evaluated according to Cochrane assessment tool for RCTs, which is the tool, and RevMan is the software that you use to analyze that.
Perfect.
Okay, and there's another one. So maybe it's about your conclusion. This is something that happened with me in a recent systematic review that I submitted to a journal, a dental journal. And my conclusion was also similar that yours. And I have written that my conclusion was that there were a lot of bias among the studies, but the suggestion was that I should write down that the conclusion should be that there was insufficient evidence to conclude that And so maybe that's the same case for you. I think you have insufficient evidence to conclude that probiotics therapy is effective as complementary treatment for patients with colorectal cancer. So maybe you should consider this as your conclusion as well, okay? Just a suggestion. You both can decide if it's going to accept it or not, okay?
Thank you.
Okay, congrats for the presentation. And Professor Andrea, the word is
Thank
yours.
you. Okay, congratulations, Fernando, and your supervisor, Carlos Eduardo, for the work, for the study. I have the same question about your conclusions of something different, but the means is the same that Professor Evelise put here for you, because I ask you if there's difference between your research and others to correlate the pyrexemia duration. It's because of the probiotics or other treatment or others, I don't know, bias. So maybe you needed to be sure that the conclusion is correct, only this. But the work, it's amazing for a student for initiating the research file. So congratulations again, and continues in this work successfully.
Thank you very much.
Sorry. Now the third presentation is by student Arielle Barreto Hagsma, supervised by Professor Cristina Pellegrino Baena.
Hello, my name is Arielle Barreto Hagsma, and I'm a medical school graduate from the Pontifical Catholic University of Paraná. Today, I will be talking about my research project, which is a longitudinal analysis between hand grip strength and cognitive function during aging, using the survey of health, aging, and retirement in Europe, also known as SHARE. This project was an international collaboration with the Central University of Catalonia. To start off, we need to understand that aging is a natural process that no physiological system is spared of, including the cardiovascular system, the musculoskeletal system, and also the neurological system. Today, it is estimated that about 40% of the elderly population of the world has some type of cognitive decline. However, this cognitive decline has always been known to be of difficult diagnosis due to its complex clinical presentations and pathological ways. Some of these complex pathological ways are oxidative stress, hypercortisolism, and neuronal loss. If we pay attention closely, we notice that these three possible pathways are also the pathways for another pathology that occurs during aging, which is muscle loss. Knowing about these shared pathways between pathologies, many researchers started studying about a possible association between cognitive decline and loss of muscle strength. That way, they believe they could use the practical equipment called dynamometer that measures hand grip strength in kilograms to identify early loss of muscle and subsequent cognitive decline. In other words, they are trying to find a more objective and practical way to identify cognitive loss. So our objective was to evaluate the longitudinal association between the loss of hand grip strength and decline in cognitive performance in different cognitive domains in the multi-centered European population of SHARE. But what is SHARE exactly that I speak so much of? So SHARE is a multinational prospective study that started way back in 2004, and until today has gathered longitudinal information of more than 140,000 individuals aged 50 or more from 28 different European countries. They conduct in-depth interviews every 2 years to assess the effects of health, social, economic, and environmental policies in aging. The project was approved by the Ethics Council of Max Planck Society and is constantly being reassessed. For our variables, we used the 3 different cognitive domains that were assessed inside the SHARE questionnaire, which were verbal fluency, verbal recall, which is divided into first immediate recall and second recall, and numeracy, while the hand grip strength values were divided into quintiles that were created by us and stratified by sex, so we could assess the effect of gender in the study. From all the 8 waves of share, we chose wave number 5 as our baseline wave and decided to study these individuals for the next 2 subsequent waves, totalizing 3 observations of each individual over the period of 4 years. Our exclusion criteria were lack of data, lack of follow-up, dementia diagnosis on the baseline, Parkinson diagnosis along the study. In the end, our population was made up of 8,236 individuals that were followed during 4 years. For our analysis, we decided to use generalized estimated equations, also known as GE. For a better understanding of what is GE, here's a representation of our study. Each color represents the 3 different observations made over time. What the GE does is to enable not only the intragroup analysis, but also the intergroup analysis of repeated measures over time of the same individual, along with incorporating confounding variables, something that other statistical methods are not able to replicate. 4 sets of analyses were made, 1 for each cognitive score. In each analysis, hand grip strength was used as a predictor for cognitive decline, and the dependent variables were the different cognitive scores. The analysis was adjusted for 6 different models, which included baseline hand grip strength, age, time, education, behavioral risks, medical records, and functionality scores. So, for our results, our final analysis included 55% of female individuals with a mean age of 67, while the male individuals' mean age was 68. We observed that individuals of both sexes in the lowest quintiles of hand grip strength, the weaker ones, were also older. They had lower educational levels, lower probability of being obese, ever having smoked or consumed alcohol. They also had more chronic diseases, more depression, more physical limitations, and most importantly, they had lower cognitive function in all the 3 different study domains. So weaker individuals had lower cognitive function. Furthermore, we can clearly see here in the graphs that individuals with higher values of hand grip strength on the baseline wave, which is the red line here, demonstrated higher cognitive scores on the baseline and less risk of cognitive decline over a period of 4 years when compared to the weaker individuals, the blue line, which presented lower cognitive scores on the baseline and more risk of cognitive decline over the years. In the longitudinal analysis, using generalized estimated equations, we observed a significant association between hand grip strength and subsequent cognitive decline in all the 3 cognitive domains, even after all the adjustments. In other words, stronger individuals had greater dose response of hand grip strength over the risk reduction of cognitive decline. For instance, a female individual in the 5th quintile of hand grip strength, the stronger one, had 3 times the increment in the verbal fluency score than a female on the 2nd quintile would have over the years. That is, stronger individuals had less risk of decline over the years when compared to the weaker ones. And this is applicable for both sexes in all 3 cognitive domains that were studied. Now we have to acknowledge some limitations of our study. The first one being that most covariates were self-reported by the respondents, thus subject to biases. Additionally, biological factors such as interleukin-6, widely known to be significant in the development of cognitive impairment and dementia, were not available in SHARE. However, many were the conclusions of our study that can help the scientific community today. The first conclusion of our study was that hand grip strength is significantly associated with cognitive decline. So it can be used as a predictor for subsequent cognitive decline in people age 50 or more. This will help in the creation of a low-cost, objective, and practical tool using the hand grip strength measured by the dynamometer to predict future cognitive decline in aging individuals, making the diagnosis of cognitive decline easier and more efficient, and also enabling prevention even from the early stages of cognitive loss. Furthermore, our study was one of the first ones to show that the relationship between hand grip strength and cognitive decline varies amongst different cognitive domains and in between sexes. This shows the need for the individualization of cutoff points and cognitive prevention adjusting them to different cognitive domains and genders, something that has been rarely done in other studies until now. On top of being one of the most complete studies on the subject, our research enables future studies to explore the impact of hand grip strength improvement and cognitive improvement in aging people. With that in mind, our study is under review in the Maturitas Journal, the International Multidisciplinary Scientific Journal of Midlife Health, with an impact factor of 4.32. Thank you for the attention, and I hope everyone enjoyed the presentation. I'm happy to answer any questions now.
Okay, Arielle. Thanks for the presentation. Professor Andrea, you want to start this time?
Okay. Congratulations, Arielle. It's a very interesting study. With a beautiful design, and use tools that I didn't have no knowledge about after reading your abstract and now seeing your presentation. So it's amazing. Thank you for this. I believe that my own question is not a question, it's a reflection about your results and the translation of these results with the clinical practice. Do you have an idea what it would be understanding or the new guidelines that the professionals can use in clinical practical using your results? I don't know if these results already have a publication, but I believe that Christine, your supervisor, will publish this. So my question is, what you're thinking about to use these results in clinical practice?
Okay, our results can be used in clinical practice because, for example, we analyzed all the data and we see that we actually saw how much the quantity of which cognitive score raises or lower because of the hand grip strength. So what, for example, it's an idea. Our study can help in the creation of a specific scale of cognitive decline in which every kilogram of the hand grip strength that you lose or that you gain will correspond to a different level of cognitive decline. So that will be one of the clinical practice that can be used, but there are many.
Okay, you show us that in the population have some limitations, and I believe that I hear you saying that the locomotion or something in I needed to remember this. I don't believe it, but okay, it's an older population. Okay, it's in the older population you have a loss of mass muscle. And this correlation with cognition is very important because you can organize it very easy equipment to measure this and qualify the cognition. So maybe the clinical practical can use this before the loss of cognition start or something. So again, Arielle, thank you for your presentation. And also I can highlight something that your presentation was very clear because it's complex. And the skills that you do with the first observation, second, and all the analysis that you made was amazing. So it was very, very easy for us to understand. So congratulations again. Thank you.
Thank you. Thank you.
Yeah, we don't have too much time, but I totally agree with Professor Andrea. It's a beautiful work. It's a beautiful study. I was just amazed how this this database can help everybody around the world that work with elderly to do a lot of things. I can imagine how many things you were just thinking about researching with this huge
there's
amount
many
of
data.
data. It's incredible. I wish I had a dental database like that for me. It's amazing. So I just wanted to congratulate you, Arielle and Chris, for this study. I wish you publish as soon as possible with a minor revision, with all that we ask for the reviewers. And I think it's a beautiful work that can further ask for another and other researchers. So it's amazing how Europeans and people around the world has this kind of idea to monitor these patients. And I think the most important thing is that it's a beautiful study, but you pointed out an important limitation, which is the questionnaires that the patients have to answer. So once it is self-reported, you have to be aware that some of these answers might not be so accurate. But any of these, any study has some limitations, and you pointed out brilliantly all the advantages and all the data that you can use with this kind of database. So congrats again, and wish you good luck with the publication. Okay.
Thank you. Thank you very much.
Okay. Yeah, we're just on time. So next presentation is student is Tatiana de Avila Miguel, and the supervisor is Professor Emanuel Góes Jr. from Med School, PIBIC with scholarship from PUC.
Good morning, everyone. Can you hear me?
Yes, Tatiana, you can go ahead. Yes.
Okay. So my name is Tatiana. My supervisor is Dr. Emanuel Góes Jr. And our research project was a systematic review for the treatment of severe diverticulitis with damage control surgery. Acute diverticulitis accounts for approximately 300,000 admissions per year in the US. The severe cases require surgical intervention since complications are frequent, especially Hinch stages 3 and 4 that have a high risk of mortality. Here we have the Hinch classification for acute diverticulitis, and the stage 3 is purulent peritonitis, and stage 4 is fecal peritonitis. Although Hartmann's procedure is considered safe for treatment acute complicated diverticulitis, the ideal surgical approach has not been yet established. Damage control surgery is mostly used in trauma, but it has been considered as an interesting alternative for treating other diseases, such as severe sepsis secondary to purulent or fecal peritonitis, resulting from the complicated acute diverticulitis. The rationale for using this strategy is that it decreases the chances of the patient having acidosis, hypothermia, and coagulopathies, because it comprises, as you can see here on the side, two surgical approaches separately. The first stage is an abbreviated approach that aims to achieve homeostasis and drainage of the septic contamination. Then the patient is allocated at an intensive care unit for correcting hypothermia, acidosis, coagulopathy, and any other conditions he may have. After 24 to 48 hours, the final surgery is then performed with full gastrointestinal repair. Currently, this strategy is reserved for unstable patients with perforated diverticulitis, but a lot of studies, and mainly observational studies, suggest that other benefits, such as the reduction in the rate of permanent stoma limited surgical trauma, and effective elimination of the infectious focus can also be obtained. However, the literature still lacks clear information about the potential benefits for treating this condition of severe diverticulitis with damage control surgery. Hence, our objective was to conduct a systematic review of the use of damage control strategy in the surgical treatment of complicated severe diverticulitis. Our research question was elaborated in the PICO format, P being population patients with peritonitis due to complicated diverticulitis. The intervention is damage control surgery. The comparator was a conventional surgical approach, and outcome was prognosis. As method, we researched databases Embase, PubMed, Lilac, Cielo, Science Direct, and Medline from a time frame from 1978 to 2021. The inclusion criteria were containing the keywords used in the research and being a randomized controlled trial. And the exclusion criteria were any study that had a scope different from what we desired, any other format of studies such as observational studies, reviews, book chapters, publications in conference, etc., and trauma surgeries. The selected papers were then submitted to data extraction and then evaluated for methodological quality using the Cochrane tool risk of bias. Here we can see that the initial search yielded 479 papers, and after 3 exclusions and exclusion of duplicates, we ended up having only 1 study that met all the inclusion criteria that we established. This is the study that was selected. It's a randomized controlled trial published in 2020. And the results of this study is that using damage control surgery enhances reconstruction of bowel continuity and can reduce the stoma rate at discharge. And these results are also consistent with other observational studies. Here we have a table that allows us to see the evaluation for the risk of bias. Some biases, as selection bias, could not be determined because the authors did not mention allocation, concealment, or randomization procedures. Performance bias and detection bias risk were high because there was no blinding. It was an open label study. Attrition bias and reporting bias risk were low. So what is the problem of all that? Is that biases compromise the internal validity of the study. They can overestimate the observed effect. And another problem of the study is that to achieve a statistical power of 80%, they calculated they needed 70 participants. However, this study only was conducted with only 21 participants, which increases the chances of type 2 errors. They also used convenience sampling, which reduces the representativeness of the study population, impacting the external validation of data. So as final considerations, this review highlighted the lack of RCTs to evaluate the effects of damage control surgery in the treatment of complicated acute diverticulitis. And the current level of scientific evidence does not allow to conclude that damage control surgery can in fact be the best strategy for managing complicated acute diverticulitis. So it is crucial that more randomized, control, and comparative clinical studies are carried out. And we plan to conduct an RCT after the results of this review, maybe next year. And here are our information for contact, and thank you very much.
Okay. Thank you, Tatiana, for your presentation. I have only one or maybe two questions for you and your supervisor. Yeah, it calls our attention that you had only one article that met your inclusion criteria. And I'm not from the med field, but I think that maybe it's because RCTs are too difficult to perform, or since it involves surgical treatment, and in a severe case like the diverticulitis. So, and it caught my attention as well, how these authors of this unique article use convenience sample in his study. Can you explain that?
You mean what is convenience sampling?
Yeah, in this case.
Yes, they just used any patients that were available in the institution they worked. So they did not try to include patients from elsewhere or to recruit patients to establish a more representative population. They only used the cases that got into their institution. It's
So
commonly
maybe,
used in clinical research, but it does decrease external validity.
okay, yeah, but maybe this is a drawback of this study, that, and you pointed out that it was, or not?
Yes, it is. It is a drawback. I don't think it's the greatest drawback. I think that the biggest problem they had was that they calculated they needed 70 patients and they only performed the study with 21 patients. And I think that's the worst thing that happened in that RCT.
Okay. And then you said that you're planning to conduct an RCT, you and your staff and your supervisor. I think it's a good idea, since it's something that it's not published currently. And maybe I suggest another PICO question, maybe in the same topic, or with a different treatment or something like that. It's just a suggestion. I don't know if it has some coherence in what I'm suggesting, but maybe it's I think it's an important and severe condition, and maybe you should try to change your PICO question, and then maybe you can publish another systematic review on this topic. Okay.
Okay, thank you for a suggestion.
Okay, congrats again. And Professor Andrea?
Okay, congratulations, Tatiana. I believe that my question was answered in your suggestion for RCT here with your supervisor and all the team, because my question would be this technique is used in Brazil already. So it's approved something. I don't know if the Anvisa or other medical classes needed to approve this kind of surgery to apply it here in Brazil. But now you answered that you are planning, and all the team are planning to do a RCT here. So this is important to analyze the efficacy and safety of this kind of surgery. So congratulations again. I agree with the Professor Evelise. Maybe change your PICO. You can publish something in the subject that could be relevant for all researchers in this area. Thank you.
Okay, thank you.
Okay. So next one is, the student is Lucas Baena Carstens, supervised by Professor Lúcia de Noronha.
Well, hello there. I would like to clarify that today Professor Lúcia is being represented by Dr. Kleber, because she wasn't able to be present at this time because she's teaching a class that could not be switched in times. But I'm very well accompanied here. Dr. Kleber has worked with me So I'll just share my screen and can you see my screen?
Yeah.
All right. So hello, one and everyone. My name is Lucas Baena Carstens. I'm a 4th year medical student here at PUCPR. I will be talking to you a little bit about MBL lung tissue expression in COVID-19 postmortem biopsies and compare them to other pandemic viruses. So my professor orienter was Dr. Lucia de Noronha. And well, the pandemic has killed over 4 million people and still poses various challenges to scientists and health professionals all over the world. We still do not understand fully how COVID-19 works, but literature already describes it as a systemic disproportionate inflammatory response associated with a prothrombotic state. I mean, we've seen it all over the news so far, but we need to understand it a little bit better. So in order to understand what we'll be talking in here, I'll take you through a few basic concepts. And first of all, I would like to walk you through the complement cascade. Well, the complement cascade is a part of our first response, our first immune response, so our innate immune response. And we'll be looking specifically at the lectin pathway, which is activated by MBL. So as we can see, the complement cascade, what it does is it recruits leukocytes, monocytes, neutrophils, basically immune system cells. It will allow the cells to phagocyte, meaning to eat, if you will, in a simplified manner, these antigens that are in our body, so bacteria, viruses, and they will also form membrane attack complexes in bacteria that are attacking us. They are the first line, a part of the first line of defense of our body. And what are menin binding lectins, or MBL for short? So in this picture, it is basically a protein that will bind itself to a menin, which is a saccharide, so a kind of sugar. MBL, they are a class of protein that will bind itself to menins, and menins are a subpart of monosaccharides, and they are present in both viral and bacterial membranes. They also interact with our own host cell membrane, so with our cells, to modulate inflammatory response, and they can activate both the complement system and the coagulation cascade. So this is an important information to have. And opsonization. Well, opsonization is a process through which the body tags an antigen or an invader with an antibody. And that will allow, as we saw before in the complement system, that macrophages will effectively phagocyte the resulting antibody-antigen complex. So this is an example, a visual representation of antibodies binding to the surface of a virus, for example, which is what we see in COVID-19 or H1N1 influenza, as we saw in 2009. But it's important to know also that this is a bridge from the innate immune response, so from our first response, to the adaptive response, which is when we start to produce specific antibodies, for that invader virus, bacteria, fungus, whatever you will. So we'll also see leukocyte signaling, so white cell signaling and macrophage migration in COVID-19. We'll see that COVID-19 shows a very specific interleukin profile. I'll highlight right now interleukin-1 beta, interleukin-4, TNF alpha, and we'll see that lungs, we observe that lungs infected by COVID-19 have a huge activation of mast cells. So that is also an important information to have. So pneumocytes will secrete interleukins, which are proteins that will activate our endothelium, promote macrophage migration, start host immune response. This immune response will then be followed by more macrophage recruiting and mast cell activation and more cytokine release that will form a loop and will release lots of cytokines. And as the infection progresses, more specialized cells migrate to the pulmonary tissue, and your immune profile can be determined as to whether you're going to have a severe case or a mild case. In this study, what we aimed to do was to compare tissue expression of MBL in lung postmortem biopsies performed in patients that died from COVID-19, from influenza H1N1 in the pandemics of 2009. And we also aim to analyze possible MBL action mechanisms that may take place in COVID-19 pathophysiology, because we still don't know exactly what is happening. So what we did is we had 3 groups. We had the first group with 24 patients that died from COVID-19. We had 11 patients that had died from H1N1 in 2009, and we had 11 patients that died from non-pulmonary, and that was our negative control group. So this, what you see here is the image of a slide that was stained with an anti-MBL stain. All the samples were collected according to all relevant National Ethics Committee's rules. Lung samples were obtained through a mini thoracotomy, so it was a minimally invasive surgery, and they were blocked in paraffin and stained with an anti-MBL. So what we see in brown here is the MBL expressing cells, and this would be non-expressing tissue. After we digitalized those images and we analyzed them through ImagePro Plus software, and what we found was something like this. Well, when we compared COVID-19 to H1N1 MBL expression patterns, we could see that COVID-19 had a higher expression and that was statistically significant, although close to 0.5, it was still significant. What we saw when we compared to the control group was that COVID-19 groups had a expressively bigger expression of MBL. But it is also important to note that H1N1 groups had also a higher expression. So what we see, we ended up with this graph here representing the expression intensity. But let's take a look to the pathology and pathophysiology of both diseases. So H1N1 interleukin profile is already described as an overexpression of interleukin 17, and your lungs will get flooded with inflammatory cells. Whereas in COVID-19, what we see is that our lungs get fibrotic, and that is due to a interleukin-1 beta and TNF-alpha secretion. And so there are different lesions that will lead to different kinds of pneumonia, is what I mean. And what's important to know is that MBL is a known upregulator for both those responses. So both H1N1 and SARS-CoV-2 infections are potentialized by the MBL expression. Now, we have a few possible mechanisms that we propose. And one of them is that MBL may be a first signal recognized by the inflammation pathway, and we'll talk a little bit more about that in the future. Well, MBL increased expression could lead to a disproportionate complement system activation, so our first responders don't stop responding, and that can be very harmful because that can cause kidney failure, that can cause vascular dysfunctions. And we can see also that MBL may work as a coagulation cascade initiator, and that is due to its activation of C5 factor in the complement cascade that can also activate factor 8. That is a factor in the coagulation cascade, so that allows your body to create microthrombi and create ischemic strokes, ischemic events in the heart, and which is something that we've seen a lot with COVID. And although it remains unsure, in this case, MBL may be a first signal recognized by the inflammation pathway, and it is surely known a normal inflammatory upregulator. And the MBL expression serves as an example of the complexity of SARS-CoV-2 infection because, as we see, it acts by recruiting cells, it acts by creating thrombi, it acts by consuming our complement system, and it acts by stimulating cells' metabolisms to produce interleukins that create a cytokine storm. That is the great problem we face with COVID. And the growing body of evidence in literature towards diverse immune mechanisms involved raise a wide array of questions that still warrant for further investigations. And as a wise woman once told me in science, the questions you ask are as important as the answers you find. And although we found some very interesting answers in this work, it leaves me with many more unasked questions, and very good ones at that. So we did a lot of work, not only looking at MBL, but as with other cytokines, other proteins as well, and we published quite a few of them. So we published a description of the cytokine storm in pulmonary tissue. We were able to publish the lung remodeling pathway of COVID-19, and we also were able to describe the disease as it acts in the heart. And I'm so glad to announce that this paper was accepted and is soon to be published in Frontiers in Immunology. And it also pointed towards inflammation activation, which is a question I want to be looking further into in the future. These were the references I used to make this presentation, but I would like to take a moment to thank the professors that worked with me, my colleagues in PUCPR's experimental pathology laboratory, and they allowing me to work with them, but especially I would like to acknowledge all the COVID-19 victims and their families, and thank especially those that were so kind as to allow us to perform the studies, and praise those frontline workers that gave up so much so that we could be here today having this talk in much better times. Thank you very much.
Thank you, Lucas. Professor Andrea, if you would like to start.
Okay, thank you. It's a pleasure to be here today, not only to see you, Lucas, but also to know some faces that I didn't know it in your group in COVID biopsies. It's amazing your knowledge about immune response, because I know that it's a very intriguing and complex pathways. And for the other side, I know that MBL and also the complement system cascade, it's very related with severity of the disease. And when I look at your abstract and I saw the possible mechanism action, I thought, oh, it's a pretty complex aim that they want to know about a piece of immune response, that it's MBL. But when you construct your ideas and you put the image, I understand the mechanisms. It's not only MBL, it's a lot of molecules that integrate with this and create a model. And this model is correlated with severity. This is very important to everybody knows here. It's not only MBL. My question is because you said something about mast cell degranulation, that it's important, and I will talking about this, and you forgot to talk about this. What is this relation between complement system molecules, MBL in particularly, and mast cell degranulation?
Well, thank you for your excellent question. First of all, I would like to, well, thank you for the opportunity, but let's go. I haven't particularly worked in the mast cell degranulation group, but I have accompanied a bit of it. And what struck me as important was that the interleukin profile that was expressed, it should have been a huge macrophage recruitment profile, but what we saw and what was surprising was that actually what we saw was that the native mast cells in the lungs were degranulating, and that was not at all expected. So it is important to illustrate the fact that COVID-19's immune response is not at all what we expect it to be and what we initially thought it to be. It will also differentiate it, for example, from the H1N1 pandemic, in which we didn't see mast cells degranulating. What we saw was an actual inflammatory infiltrate in the lung that was basically flooding those alveoli and avoiding. And that mast cell degranulation will then, how can I say it? They will start the process of fibrosis in the lung through the interleukin-4 secretion that they have, that was plenty marked, and of course that was not in the MBL portion of my work. But as I was lucky enough to be able to work with so many groups during this time, I got to see a little bit of that. So I hope my answer was adequate and up to your expectation.
Yes, it's true. It's okay, and just want to said something about your presentation that was very, very for me, because I have created this image of all the immune system in my mind about this disease. So it's very illustrative, and using these figures is very important for students to understand. So was a great presentation. I would like to have more time to talk more about everything, Lucas, but it's not the way. So I will pass the word for Professor Evelise, and thank you again and congratulations.
Thank you very much. I'm honored.
Thank you, Andrea. And yeah, we indeed, we don't have too much time, but I want to congratulate Lucas and all the team involved in this study, and I can see that a lot more studies and publications that will come after that. And I have to say that you gave us an immunology class here today, and I'm not a dental student. I mean, I'm a med student, but I also have to say that I understood a lot what you showed us today. So it seems like you're a kind of native professor English. Your English it's amazing, and in your fluency and everything, and you showed us in a very simple way and something that is very complex. So thank you for giving us this all this explanation, and you used quite well all your time. You had 10 minutes and you finished just sharp in 10 minutes. The only thing that I wanted to ask, although the study is already being submitted and received awards and everything, but you showed us a p-value for COVID was almost not significant when compared to H1N1.
Yes, of course.
And you pointed out.
Yes.
So you think sampling will influence in this p-value or not?
Well, yes and no. We have a reasonable sample as to working with COVID. 24 patients to undergo a thoracotomy is not a very simple procedure to do. But what I wanted to highlight is with MBL, as I said before, it is very expressive, and it's an upregulator of both H1N1 and COVID-19. So they're different diseases, yes, and but what differentiates them is the bigger picture. It's not only the MBL, because MBL will make your inflammatory cytokines go up as a general rule, but it's what kind of interleukin, so if it's 1 beta or 17, for example, that will determine what kind of lesion will result of that infection. Because MBL is a very, it's a protein that comes in the very beginning of your immune response. So it will only start the differentiation of your response, and what cells and what interleukins and what metabolism will be involved, then that will define what is. So yes, I could expect a higher sample would give me a more significant P, but at the same time, we need to look at both diseases as increased expression of MBL.
Mm-hmm. Okay, maybe something that a reviewer would comment or suggest.
Thank you very much. It was an excellent question.
Yeah, you should be prepared to answer that. Okay, congratulations again.
Thank you very much.
It's a very relevant scientific, and I mean, just have to wish good luck for further experiments and studies that you and your team are going to perform, okay?
Thank you very much. It was a pleasure.
Thank you. Next one is Gabriel Silva Moraes, supervised by Professor Marcelo Távora Mira. Good morning, Gabriel.
Good morning.
And
Just
you
share
can
my
start.
screen. Can you see it?
Yeah.
Okay, great. So, hello everyone. My name is Gabriel Moraes, and I'm a second-year pharmacy student advised by Professor Marcelo Távora Mira, PhD. This study was entitled Association Analysis between AGO, EGR2, and CDH18 exonic variants and leprosy, funded by Fundação Araucária. As a background, leprosy, also known as Hansen's disease, is a chronic infectious disease caused by Mycobacterium leprae. It's an acid-fast bacillus, which infects mainly Schwann cells and macrophages. Leprosy was the first human disease to which an infectious agent has been described. It happened in 1873 by Gerhard Hansen. Leprosy is highly curable with multi-drug therapy, but untreated, it can cause progressive and permanent nerve damage, skin damage, or limbs damage. This is an example of the ulnar nerve damage that prevents from the people of closing their hands properly, and we call this kind of aspect as claw hand because of the aspect. Although the multi-drug therapy has decreased the number of new cases yearly from leprosy, it is still a problem. Over 127,000 new cases are reported globally, and among these, almost 18,000 comes from Brazil. Mycobacterium leprae has a quite long incubation time, and it cannot be cultivated in artificial media. Its clonal structure worldwide, added to observational studies and new molecular methods, stimulate the understanding of how the host genetics is associated as important factor in leprosy development. Leprosy's diagnosis is essentially clinical, and the exposure to Mycobacterium leprae is necessary, but not enough to cause the clinical disease. Inside the clinical disease, we have a spectrum of clinical forms that can be classified in two major systems. There is a system proposed by Ridley Joplin in 1966, and it classifies the disease according to the immune response from the host. So there is a tuberculoid polio with a majority of cell-mediated immune response, and a lepromatosis polio with a humoral immune response. And among these two polos, there are three borderline forms. Also, there is the classification from the World Health Organization. They classify according to the number of skin lesions, and they classify the individuals as paucibacillary or multibacillary. When an individual cannot be fit in one of these boxes, we call it, and we say it has an indeterminate leprosy, and the disease itself, regardless of the clinical form, it's called Hansen leprosy per se. As we said, the M. leprae has its genome structure conserved through the years, and it's basically clonal around the world. The genetic factors associated with these mechanisms are attributed to the host. GWAS, or genome-wide association studies, are approaches that involves rapidly scanning markers across the complete sets of DNA of many people to find genetic variations associated with particular disease. These studies are important to find general associations with the study disease. And I want to highlight this leprosy GWAS today because it discovered 6 new susceptibility loci, and among these, the three genes in focus of this study. Nature published in 2008 a news feature that was about the case of the missing heritability. It was about the thought of the scientists that with GWAS, we were supposed to develop and know everything about genetics associated diseases, but that was not what happened. Because GWAS have some kind of limitations. To perform a GWAS, we need massive sample sizes, and as a result, we get common variants, associations, low odds ratio, and principally, it misses a huge part of the heritability. So where does this heritability come from? The hypothesis is that the signs viewed by the GWAS in these common variants are actually in strong linkage disequilibrium with rare variants that are missed from the GWAS. We need to find another approach to focus on these rare variants, and that's exactly what we did on this study. We performed a direct sequencing approach to find these rare variants. And with that, the objective of this study lies at the comprehension of the genetic control in the host susceptibility to leprosy, applying association analysis between the disease and the variants positioned in translated and untranslated region of the AGO, EGR2, and CDH18 genes. This is a family-based association study, and 177 blood samples were recruited from families living in Vila Santo Antônio do Prata. It's in north of Brazil. This village is a former colony of leprosy, and it is still hyperendemic and isolated for the disease to this day. DNA was extracted, followed by DNA quantification, and next generation sequencing was performed using Illumina HiSeq platform. The raw data from the sequencing was first aligned using a genome of reference using the software Burrows-Wheeler Aligner, and then filtered. After that, the variant analysis was performed. Since the association test, we used the FBAT software, that is family-based association test, and also the transmission disequilibrium test. For odds ratio, we calculated using the SAS software. For leakage disequilibrium, we used HaploView, and variance impact were used the HaploReg. As results, the content, the mean content of GC content was around 44%, which was expected. And this analysis was made to be sure that our sequencing was in a great quality and accuracy. Also, most of the reads have an average quality greater than Q30 in the Fred scale, which means that the reads have an accuracy greater than 99.9%. So we found 28 variants with minor allele frequency greater than 0.04, and these variants were identified in at least 8 informative families. From these variants, the transmission disequilibrium test revealed one association with leprosy per se, which means the disease itself, regardless of the clinical form. This variant is located in the untranslated region of the ADO gene and has an odds ratio of 5.53, which means that an individual carrying this variant is like 5.53 times likely more likely to develop the disease than an individual not carrying it. And the p-value was statistically relevant. Also, we found 103 variants with minor allele frequency less than 0.03, which classifies them as rare or low frequency. And the linkage disequilibrium analysis between the variant associated in this study and a variant associated in previous studies was also performed. In this linkage disequilibrium, we didn't find significant results in 2 Asian populations, and further studies could be more clarifying for that information. As conclusion, we identified a variant with potential impact in the ADO gene expression as predicted in silico, and we validated the association between the host genetic susceptibility to leprosy and the ADO gene. As I said, the results obtained from this study could be further validated in populations with a distinct genetic background or in larger populations. That concludes my presentation, and I must thank at first Professor Marcelo for the opportunity and my lab colleagues Andressa and Gia. Thank you all for your attention.
Thank you, Gabriel, for your presentation. Professor Andrea, if you want to start this time.
Okay. Thank you, and congratulations, Gabriel. Congratulations, Marcelo. Yesterday, I saw another part of this work with Gian. I believe that it was the student. It was very good presentation, and also Gabriel. Thank you for your presentation. It was amazing. I'm searching something because I read the abstract first from this section. And I searched something about ADO gene and the protein that it's translated by this gene. And I saw that they have correlated, it's not a very expression in normal tissue, but it's increased expression in some cancers, especially in liver cancer.
Yes.
I don't know if, yes, okay, so I searched correctly. I don't know. My question is, the relation between cancer and leprosy, it's already being studied for such investigators, but in the population of Prata, you have some correlation with the cancer appearance in this population or not? No?
Okay,
No,
thank you for your
Marcelo is
question.
doing this.
Marcelo already answered it.
Yes.
As you said, the AGO gene is not usually expressed in normal cells. It is commonly expressed in leukemia cells, in hairy cell leukemia, and also a gut cancer, as you mentioned it. So we don't have access to any cancer data from the Vila Santo Antônio do Prata. So we cannot make these associations with leprosy and cancer in this specific population.
Okay, so now I know that Marcelo's team is developing some cells to organize the functional assays into the lab to understand the functional impact of these genes or these variants in leprosy susceptibility. You can organize in your mind some steps that you can induce it using these protocols or these approaches to understand the impact of this variant.
Sure. As I don't know if you had this information, but I'm actually working in functional studies with Marcelo for the for the validation of this association between the epidemiological genetics and the cells and the functional studies. So yes, we are working with cell culturing and cell reprogramming. So our goal is to patronize some studies, some experiments to make these cells viable for some stimuli with the Mycobacterium leprae. And after we know how the cells respond to Mycobacterium leprae with the variant, without the variant, or how it stimulates in reactive oxygen species or apoptosis or these natural cells events, we can answer better how does this variant impacts the functional part of the cells.
Okay, and amazing. So now I'm jealous, Marcelo. You have two wonderful boys. I wanted to bring them to my lab. So congratulations, success for your next step. Okay, and I will see you in the lab. If you need some help, I will be here. Thank you, Professor.
Thank you all.
Thank you, Andrea.
Okay, I'll be brief, okay? Gabriel, congrats for your presentation, for the brilliant study, and to Professor Marcelo as well for the huge expertise and publications on this topic, which I think they are very scientifically relevant for the topic and also for PUCI, for everything that you have done in your career. So my question for you, Gabriel, Professor Andrea, I had already said, I was about to ask you which kind of study you think you're going to do it after this one. So you already answered, and
yes, I'm already working on it.
Yeah, I'm glad that you are still working with it, and further studies will be reproduced after this one. So congrats again. Congratulations, Marcelo, for your brilliant work with these students. Okay.
Thank you.
Thank you so much.
Thank you. Yeah, we're almost there. We have two more. Next one is Nicolas Henrique Borges, supervised by Professor Marina Luiz Viola de Azevedo, PIBIC with Araucaria Foundation Scholarship. Nicholas?
Good morning. Can you see my screen?
Yes, you can go ahead.
Just 1 second. Okay, here. Hi, I'm Nicholas, and I will present my research entitled Analysis of Immunohistochemical Tissue Expression of Interleukin-8 lung samples from patients who have died from severe lung disease caused by the SARS-CoV-2 and H1N1 viruses. My advisor is Dr. Marina. I'm a medical student from Campus Curitiba, and this work was founded by Fundação Araucária. With the lack of interleukin-17 receptors in the lab, the central objectives were changed, and we ended up to use the interleukin-8 that shows the particular SAM pathway. In December of 2019, a new respiratory coronavirus emerged in China, causing a wave of infection and deaths. The World Health Organization estimated that 40.9 million people have died until today, and the viral infection can trigger an inflammatory process and release of cytokines. The H1N1 is a pandemic virus of 2009 and can release the interleukin-17 and interleukin-8, playing a role in acute lung injury. And here, there are two important glycoproteins. First, the hemagglutinin that can bind the virus to host cell receptor, and the neuraminidase that can induce the viral replication. The SARS-CoV-2 is a pandemic virus of 2019 that is a positive strain RNA virus, and this is an important point because just because of that, the virus can use the host cells like your own machine and can induce the cytokine storm with the emphasis of the interleukin-6. The virus can bind to the receptor through the protein spike or protein S and infect the human cells, causing a wave of the endothelial dysfunction, endothelial activations, and diffuse alveolar damage. The interleukin-8 is a chemoactive interleukin that can prolong the neutrophil activity and migration because they downregulation the pathways of apoptosis and pyroptosis. They can exacerbate the inflammatory response. But why the Th17 immune response? The interleukin-8 and interleukin-17A can be used for evaluate the antiviral response and can show a possible therapy targets acting in an infection, inflammation, and autoimmunity. And why the H1N1 versus the SARS-CoV-2? In 2009, the laboratory of experimental pathology analyzed the interleukin-17 and interleukin-8 and H1N1 with the thesis of Dr. Marina, and with the knowledge of other pandemics, we replicated, evaluated, and compared the same biomarkers in COVID-19. The central objectives is evaluate the tissue expression of interleukin-8 and correlate with the presence of the neutrophils in pulmonary patients, and then characterize the samples according to gender, age, and histopathological pattern of the disease. The materials and methods, the study was approved by the National Research Ethics Committee. All families sign an informed consent form, and the sample collections follow all the safety protocols. The study samples consist in 20 cases of COVID-19, 10 cases of H1N1, and 11 control cases that is formed by neoplastic process, but without neoplastic cancers in the lung. The materials and methods, the lung samples were separated in tissue microarray paraffin blocks, as you can see. And after that, the immunohistochemistry was performed using the antibody of the interleukin-8. The immunostaining slides were scanned by Axion Scan Z1, and the image was selected by randomized and blinding selection. And then the Image Pro Plus was the app used for obtaining the tissue expression of interleukin-8. And there are the neutrophil counts for hotspot zones. And for statistical analysis, the Kruskal-Wallis non-parametric test was performed. And in this image one, you can see in the black arrow showing the neutrophils, the black circle showing the alveolar septum, and the black stars show the hyaline membranes. So the diffuse alveolar damage caused by the H1N1 differs from those observed in COVID group. There are fewer hyaline membranes, increased recruitment of septal and intraalveolar neutrophils, and no significant fibrinous thromb were observed. In the table 1, the mean average in COVID group is higher than the H1N1, and the virus attacks more men than women. When you talk about the expression of interleukin A, there are 5 times more expression in the H1N1 than the COVID group, and the neutrophils looks like the same, but 7 times more. The COVID group remains more time on mechanical ventilation, and there are more time in from hospitalization to death. And the p-values shows here shows significant statistical. And our work has some limitations. First, the small sample of the evaluated group, and second, the immunohistochemic shows a static information at the time of the death, and it looks like a photograph. We can't see all frames of the infection, and we compared 2 different types of pandemic viruses. In conclusion, there are 4 important points. First, the SARS-CoV-2 and H1N1 infects the epithelial cells of the alveolar lung. 2, there are release of cytokines with the emphasis of interleukin 17A and interleukin 8. And 30, these cytokines can make a neutrophil recruitment, causing an increase of inflammatory response. So the patients infected with SARS-CoV-2 had lower tissue expression of interleukin 8 and neutrophil counts. This finding implying a possible evasion of the adaptive Th1 response, culminating in a Th2 response being less affected to the viral agent. These findings impact the mortality of the patients. These results were published in Frontiers in Immunology, and this PIBIC gave me opportunity to publish in other journals about the COVID-19. We published in the American Heart Association and described some case reports in the Oxford Rheumatology. And it described another case report in Sociedade Brasileira de Cardiologia. This is my reference and my acknowledgements. Thank you.
Okay, Nicholas, you finished?
Yeah.
Still have time. Okay. Okay, so congrats. This is a huge teamwork. I can see that you have already published papers on that, and I just have to congratulate you because your academic career is amazing already. So I only can ask how far can you go? Are you still looking for further studies with the same team on the same topic, I guess? So what's next?
Oh, I'm so proud of this team because I could decide that I want to be a researcher and still in this career. It's a great experience and a place where you can make friends and the people can really help you and send a hand like, no, I will help you. I will, I don't know what are you mean, but I'm willing to teach you. I will take a force in this, and this is so important to me. I'm so glad of the members of Biopsias Pandemia and the friends and family. This look like a family.
Good. You should be proud of yourself and of your team.
Thank you.
You are part of this team as well. So I congratulate you and your supervisor and all of those involved in all those studies that are really important. And I think improvements in treatment of COVID can arise from all this data and all these efforts that you guys have done inside the labs and the hospitals and everything. So this is huge. This is really important. Thank you for that. And congratulations again. And Andrea?
Okay, so Nicholas, great presentation. Marina, you teach so well. I'm so happy to be here, to see Marina as a supervisor and Nicholas to look your face, because I know you for the test, but not here. Your face. Now I can put an image in my mind when I look at your messages in the group. Nicholas. Okay, now I know how Nicholas is. So I'm very happy to be here because I know the efforts that this team can put during the disease. The efforts that this team put during the collection of the samples, that is not easy. It's was amazing what this team does or done during the COVID this last year and this year. I have a lot of questions, but it's not relevant by now because you have a lot of publish your results. So I wanted to, that your presentation was very clear. You put the origin of the idea using the PhD of your supervisor and the questions about this new pandemic virus. So it's very clear for the audience to follow your object, your aim, your goal, the way that you put the images and all the sequence was very important for the audience to follow you. And I'm very happy. I have some questions here, but we can discuss later. I'm just want to know that you already have an amazing opportunity and you enjoy this opportunity the best way. So thank you for your presentation. Thank you, Marina. I'm very, very proud of you. Thank you. Thank you.
Yeah, and I want to say a special thank you for Dr. Marina. We make friends, we make a family. They support me all the time, and I love them. And thank you, special thank you for my friends and my family. I love them. Thank you.
Okay, thank you, Nicholas. Thank you, Professor Marina, and congratulations for both of you. Next one is Beatriz Martins Rodrigues Carvalho de Almeida, supervised by Professor Felipe Francisco Bondantuon from Med School, and this is a PIBIC with a CNPq scholarship.
Very good. Can you guys hear me and see the presentation?
Yeah.
Okay, very good. So we'll start. So good morning, everybody. My name is Beatriz Almeida. I was advised by Dr. Felipe Tuon, and I'm a biotechnology student here at the Curitiba campus. And today I'm going to be talking to you guys a little bit about the development of human and bovine bone granules impregnated with silver nanoparticles. So bacterial adhesion and biofilm growth to both bone and teeth surfaces, and then subsequent the biofilm growth itself, is the leading cause of infections in both the orthopedic and orthodontic fields. And the biofilm production is very problematic because it helps the bacteria evade the antimicrobial agents that will be taken by the patients post-op. The problem is further complicated by the structure of the bone itself. So the bone is spongy, it's porous, which means that our immune cells don't have adequate access to these areas in order to help fight off these infections. So these two factors together lead to higher doses of more powerful antibiotics used by patients post-op. And this is a very big problem because it leads to antimicrobial resistance. So as we know in the biological area, antimicrobial resistance is one of the biggest challenges that humankind is facing now and will face in the future. So a simple Google search will return thousands of articles talking about how bad the problem will be in the future. And the World Health Organization predicts that by 2050, if we don't tackle the problem now, one person will die every 3 seconds due to complications from antimicrobial resistance. And just to put that into context a little bit, in 2018, all of the cancers combined were responsible for the deaths of 9.5 million people, which is about 1 death every 3.3 seconds. So antimicrobial resistance could become a bigger killer than all of the cancers combined. So in the orthopedic and orthodontic fields, the goal should be to develop a granular substitute for bone grafts in which the substitute contains bacteriostatic and inhibitory activity on the biofilm, as well as potentially osteogenic potential. So we looked into silver nanoparticles because in the literature, they had already been described as having very high effectiveness against microbes. So when we tried to think about this, we looked at what had already been published, and the search for bovine bones and silver and nanoparticles returned 9 results on PubMed, and none of them talked specifically about adhering nanoparticles to the bone surface. So we knew that this study would be very important for the scientific community. One thing we should talk about before talking about the methodology is that we really wanted to develop some sort of human bone as well, but we relied on the tissue bank at PUCI for the human bones, which they stopped receiving in early 2020. So we did have some bones that we used that were left over, but since we couldn't repeat the results, we decided not to include them. So the bovine bones were then procured from a local butcher. We started by drilling an area around where we wanted. We would then take a sickle and some tongs and remove a bone cylinder that would then be hammered down into a bone disc that is approximately 2 millimeters in diameter and 2 millimeters high. Here we have a bone purification process that included the removal of fat, protein, and other contaminants to make the bone as pure as possible. But I have intentionally left this blank here because we do have a patent and an article that are pending on this process. So for the methodology, the silver nanoparticles were actually produced inside of the lab using a methodology previously described in the literature. And we had certainty that our nanoparticles were formed because the solution would change from clear to yellow very distinctly. And this is due to the Tyndall effect, which is a physical effect that describes how different particles will deflect light in different ways. So the silver nanoparticles would reflect light in a yellowish manner as opposed to the molecular components that led to the production of the nanoparticles. We would then impregnate the bone discs with the nanoparticles, and this was done just by simply submerging the bones inside of the solution. And then bacterial testing was carried out. So things like analyzing halos, analyzing inhibitory activity on the biofilm, etc. So for the results, the first thing we needed to make sure was that we actually had nanoparticles and the size of the nanoparticles. So yes, we had the Tyndall effect, but we needed to confirm it further. So we see here first this very characteristic peak at approximately 420 to 430 AU in the ANISA analysis, which indicated that we had nanoparticles that were about 50 nanometers in size. And that was also confirmed in the scanning electron microscopy. So here you can very clearly see that the average size of the nanoparticles was about 50 nanometers. Besides that, the other thing we needed to confirm as well was that the nanoparticles had adhered to the bone surface. So here we see the very porous structure of the bone, obviously with the nanoparticles adhered. And we also see here the EDS analysis, which analyzes the chemical composition of the samples. So the very important thing here to see is that we have characteristic calcium and silver peaks in the same sample. The important thing being the fact that these were in the same sample, which confirmed to us that, okay, we did have the silver nanoparticles adhered to our bone surface. Here, as you can see on the right-hand side of the screen in these graphs, all of our samples had p-values under 5%. Specifically here, you can see P. aeruginosa, and E. coli, which are organisms that are more commonly known to the general population. And the p-values being under 5% indicated that we had a very big difference between the statistical group and not the statistical group, the control group and the experimental group, which means that the silver nanoparticles had a very big effect on the biofilm production, as well as the colony forming unit count, which you can see here on the y-axis. The very important thing to also talk about here is the multi-resistant A. baumannii strain. So this was the only multi-resistant strain of microorganism that we used. But as you can see, the silver nanoparticles led to a 2.7 log reduction in CFU count, which showed that the silver nanoparticles not only were very effective against microorganisms, but against multi-resistant microorganisms. Here we have the results of the cytotoxicity test. So in A and B, we have our control group, and then we have our experimental group in C and D. So around the bone here, the important thing to notice is that we have a halo, right? So the cells here have died, which does indicate some level of cytotoxicity. And then in this first monolayer after the halo, we see that the cells are morphologically different, which as compared to the control group, which also indicates some level of cytotoxicity. We have two theories for why this could have happened. The first and obvious theory being that, okay, the silver nanoparticles were cytotoxic. But the second is that in our purification process, we do need to use some reagents that are a little bit extreme to human cells. So we're thinking that maybe because of the porous structure of the bone discs, something could have been left over. And that could have been what was cytotoxic as well. So further analysis would need to be done to verify if it was specifically the nanoparticles and not something else. So for our final considerations, we have here that very promising results were obtained. So the nanoparticles were effective not just in eliminating microorganisms, but eliminating multi-resistant organisms. In the future, it would be nice to test other multi-resistant strains so that we know the scope of the nanoparticles and eliminating other multi-resistant strains. It would be nice to look into other medical areas in which silver nanoparticles could be applied as alternatives for antimicrobials, because it's very important that we as scientists always aim to eliminate the environment for which superbacteria can evolve. If that environment does not exist, if we do not give them the chance to evolve, we're already very effectively combating the problem, the huge problem that is antimicrobial resistance. The last thing is that we used a green methodology to produce the silver nanoparticles as compared to other methodologies that are very, that contribute a lot to general pollution. So we don't have to worry about solving one problem, but contributing to another. We can try to minimize damage on both ends. I would like to thank SEMIC for the stipend that they provided that made it very possible for me to be a part of the project. To my advisor, Dr. Thuon, and our entire laboratory group at the Laboratory of Emerging Infectious Diseases. To PUCI as well for always providing its students with opportunities for research. So this is my third research project at the university, and I very strongly believe that all of that has contributed to my professional career. To my parents and my sister for their continued support always, specifically a big hi to my mom, who's very proud to be here watching, and to all of my friends as well for their amazing support. This project was carried out during the height of the pandemic, and my entire family is not from Curitiba, so it was very nice to have my friends virtually supporting me and helping me through everything. And thank you so much.
Thank you, Beatriz, for your wonderful presentation. And I guess your supervisor is here as well, Professor Thuon. Congratulations for this study as well. But I'll have Andrea to talk first, and then I'll have only one question later.
Okay. So, Beatriz, thank you. Congratulations for your presentation. You have a brilliant presentation, as all the presentations today and this morning was very amazing. So, thank you. I have actually two questions. You said in the end of your presentation that maybe the silver microvesicles have some cytotoxic effect, or some products that you use to during the experimentation that can cause this cytotoxicity. You evaluated only silver in vitro cells to see if they have a concentration that can be used and not cause this cytotoxic effect?
Yeah, so in terms of just evaluating silver, I think we decided not to do that because the effects of silver and silver nanoparticles are very different, right? So we know that nanoparticles behave very differently to their macro counterparts because of the increased surface area. We didn't do any concentrations to know, because we had like one concentration, which was the concentration that was produced in the lab. But one thing that we do know, and that I've been looking into in the literature, is that the size could also affect it. So we know that the smaller nanoparticles are, the more toxic they are. So maybe that could also be something to look at in the future. Maybe we need to change the methodology a little bit to make them a little bit bigger so they're less cytotoxic, you know, because the bigger they are, the less surface area they have, so the less reactions can occur.
Okay, nice. And do you have some idea how these micro particles can inhibit the biofilm production?
Yes, so it does depend on the microorganism. In E. coli specifically, another counterpart in our lab, her name is Jaycee, she was actually a PIBIC master last year. She did analyze specific resistance genes to silver nanoparticles in E. coli. So we've seen that some of them can develop resistance genes, but we do know that the resistance genes that they develop are much less than to microbes in general. But they can do things like affecting DNA replication, they can destroy membranes, they can affect RNA production as well as ribosome production. So it does really depend on the microorganism to know the mechanism.
Okay. And last question, Evelise, and then I will finish. What would be the economic impact of using these microparticles in clinical practice? Do you have an idea? This is expensive to use, or is cheaper than the other materials or the other biomaterials that exist?
Yes, of course. So the first thing is that bovine bones are very cheap and very easy to come about, right? Human bones, not so much, but I do know that in specific transplants, we don't always need to use human bones, right? We can use things from other sources. So that's one positive. The second thing is that by eliminating the antimicrobial resistance problem, we reduce a lot of the health costs, right? So I don't know if you saw in my presentation, but one of the article titles was about how much antimicrobial resistance ends up costing the NHS, right, which is the National Health System in the UK, per year, and it reaches billions of euros. So that's one of the things. And the third thing is that it's very cheap to produce the nanoparticles. All you need is like silver nitrate, glucose, and it can be done with a very simple laboratory setup. There's nothing really expensive about it. So there's multiple positives in terms of economic gain.
Okay, brilliant. Thank you, Beatriz, and congratulations for your advisor, Tuon. Congratulations. So thank you, Evelise.
Thank you so much.
Okay. Yeah, I don't have actually a real question. I was just wondering if some dental implants also work with this kind of concern regarding infection and implant loss. Mm-hmm. But this is not my expertise, so I could really not say too much about it. But I could see that these nanoparticles that you are working with, they are not homogeneous on size.
Yes.
So you talked about that maybe these major particles could be responsible for your cytotoxicity outcome. So I guess this is the next step for you to work on.
Yeah, so it's actually like the smaller they are, the more cytotoxic they are. Our bigger particles are actually less cytotoxic. One thing that would also be very beneficial to do, there was a lot of stuff we wanted to do that we were unable to do because of the pandemic. So theoretically, we should have been able to also already test this on lab subjects, like little lab rats. And we could also have done what is called a zeta potential analysis that one determines the particle charge of the surface of the nanoparticles, and that would also give us the average size of the nanoparticles, but we were unable to do that as well. So like you said, it would be nice to go on further, since the methodology we used has already been described and validated in the literature. We assumed that we would have kind of like a standard average size, but like you said, when we look at the microscopy, we do see bigger and smaller nanoparticles. So yes, that would be something to look at in the future as well.
Yeah, that would be interesting, and then you can answer a lot of these questions, but I guess you have a lot more, and there is a field for further studies on that, and I wish you good luck with this. And I guess it's already your third research, but I guess you're gonna continue working with this, and I wish you a lot of success on this and publications as well.
Thank you. Yeah, I did. I worked with nanotech all of my college career, but I decided to do a complete 180, and I'm actually looking at a master's degree in biofuel. So I'm not going to continue, but I'm sure that there will be a lot of very competent, very amazing students that will come after me and be able to continue the work.
Sure. Yeah, that's it. So congratulations on your study. Congratulations, Professor Tuon. And I guess that's it. And I just wanted to say that it was a pleasure to be this morning with you guys, with Professor Andrea. It was a pleasure to be with you and contributing and collaborating in this examination. And for all the stuff behind us, for the audiovisual, Elena, and congratulations everyone. All you guys were just too difficult to examine and to evaluate all these studies for me, which it's not my field, but I was just amazed how those studies here were so top level. And congratulations to all the supervisors as well, and all the staff members of PIBIC and Professor Klebe for the invitation. Thank you, all of you. Andrea, you want to say something for finishing this section?
Just for finish, I would like to say for all that was a pleasure to be here. It was amazing to see how these students could be work so hard during the pandemic. So this is amazing. And I'm very glad to be invited to be here. So thank you for all. Thank you for students, and continues in this way that it will be a brilliant career for all. Thank you.
Thank you, guys. See you.